We have also developed some new statistical genetics tools, also bring in different approaches, not only genetics, and I will talk a bit about them. But first of all, I think this is quite exciting with the new kind of precision medicine approaches now emerging in all of our medicine. You probably read this paper. They are trying to build this, all of us in the US, but I think we have a lot of better opportunities for those things in the Nordic or in European scenario. But this is quite interesting, I think. How will these new data, these new approaches change medicine? And this is, I think, happening as we are talking about all kind of methods and everything out there in the clinics. This is what people will have. This is what is going to happen. And, genetics, big data, that's why we're here, big longitudinal cohorts, and everything here is what we're talking about in like a epidemiological setting. But then, of course, we have all these new tools and electronic health records and stuff. But I think what is quite important also is the privacy and trust. We'll come a bit back to that. Sensitive data, we can share it everywhere. And then we need diversity. But anybody here read this paper? The high impact paper. They didn't mention any neuropsychiatry disease at all. It was only cancer and some diabetes. So I think this is a misconception in the field that, it's in Alzheimer where we can do precision medicine. That is really, it's a highly heritable. You can do all the big data. We have all these resources. So I think there's something missing here, that they are promoting the wrong diseases. But we are not doing that in the, can we get the Nordic region? So we set up this kind of way of exploiting leveraging, the health data in the Nordic region to promote or find a way for doing discoveries, including brain diseases. And of course, you all know here, that technical revolution driving this with genetics, and at least for mental diseases where I work a lot, there's a polygenic architecture, many small effects. But I think in Alzheimer, I will show you there's this mixture of rare and the common variants that we need to take into account. And why this is going to change the healthcare is, at least in Oslo, it's cheaper. You've seen this, haven't you? Cheaper to genotype than park at the hospital. No, and it's cheap to do DNA genotyping now. And this is Alzheimer's disease. We've been involved in several of these GWAS studies. It's more like a mandatory thing. You need to bring them together, publish these data. And this was the big study, most recent now. And you see the picture now. It is a lot of variants. And then it's not only the ApoE. I totally agree that it's important and everything. But you see now the kind of all the small variants popping up. And I agree that they may not be useful for drug discovery. Where should you start in this? But they can be helpful in prediction. That's the point. And the same with Parkinson. Anybody doing Parkinson work here? We have these rare variants, wrong effects, but then you have this kind of all the small effects all over the place. So this is we've contributed to all these studies. So, but then what we've been working on lately is, okay, what's the next step? And if you're really into genetics, you say, oh, let's find more samples and have instead of 95 variants, we want to have 97 and make, But I think now we need to see, can we bring some of these discoveries into the clinic? Can we find anything? Because I think, especially in psychiatric genetics, we've been sort of selling this. Oh, we need more samples, need more funding. But people are getting a bit tired of that. You need to have impact. And one kind of impact that this is what I discussed earlier. Many of you have been on this study. Found I think there were 5 Icelanders who had this rare variant, but so it's population based, explained variance very low, but it gives you a drug target, and that's what these the micro. I think not on the market yet, but they develop a lot of new studies now based on this. And what we have been working on is more like an analytical approach. I don't want to go that much into it, but with all the new data here, I think that is the big data approach. You can start doing modeling. We have hundreds of thousands of variants from millions of people. So I guess some of you do know the infinitesimal model. All markers have an effect, but we do this mixture model. And so if you have all the SNPs here, the numbers on the scale here, and then you can effect sizes. These are the Bonferroni SNPs. And here we see the mixture of effects with the null and non-null effects with mixed effect directions. And that's the concept. That you can apply this to better prediction. Anyway, so how can you identify then late onset AD cases? Because if you already start having memory, then it's too late. Even the best drug is not working. So what we have identified, what I talked about, all the variants with APOE beyond the rare variants. And then, should we use machine learning or this and that? You can train these models some sort of by supervised machine learning. You don't throw everything in, but there is a lot of knowledge in the field already. You can tell the sort of machine learning. I'm not a sort of computers expert, but the concept. And then you need to ask the right questions. I think that's the point. And I would say it's not if you get Alzheimer, but when. And so a lot of the studies now, GWAS, everything is based on case control status. But if I think this is more like a tool from epidemiology using like hazard scores. So we have developed this kind of polygenic hazard score, taking all the variants into account. And then you get the classical survival curve. This is trained on the ADGC, the US sample. And here, everybody is healthy, no Alzheimer. And here you see the age of onset. And we define them based on, all these variants, capturing also the APOE. And you can see 50% here with the worst. And you can also see the difference between the predicted and the real data here. So it's quite a good spread. And then 50% here, survival or developed when it was 75. And then the highest risk is 95. So it's quite a large spread in age of onset. And at least to me or my patients, or, they want to know if you get the disease, if you're 75 or 95. That's a pretty important information. And I know this kind of competing risk and everything that's been tested in these models and everything. So the point is that and then you can develop these, the peak age of onset. And here we can see these common variants tend to affect age distribution beyond APOE variants. So you could say that there are some, how do you say, they limit or they promote or they interact with the rare variants. And you can then and this is just to show, since ApoE was a topic today, that this is just beyond ApoE, that you can see a strong effect of this kind of the forest of all the small effects of the small houses on the Manhattan plot, that there is they are increasing or reducing the effect beyond ApoE. Anyway, but then, so why? And I think this is important clinically, that you can then, these plots here are relative estimates, but then you can put them into absolute risk if you know that in the population. And here, of course, how you use this, and there are a lot of ethical and problems in doing this, but you can see that there are large variation. If you have 80-year spins and you have a certain percentile, you go from like 0.3 in the percent of Alzheimer's disease to 10.5. So it's a huge difference in risk, in absolute risk. So then we tested the model in the Nordic countries. It's working there also, as you can see. But so why should we do it in Norway? Because the reason is we there have really good epidemiology data. And this is, I don't know, anyone here knows Trøndelag. It's called the Hunt study. This region of the country follow very closely. And like 10,000 have now been screened in a big screening study, everybody beyond or above 70 years. And you see there are like 900 that's reading, and I think many of them are these centenarians that go beyond 100. So there's a lot of opportunities here. But my point was then you can really get the very precise estimates of risk. And I think the Norwegian government now is using these numbers to estimate the need for healthcare the next years in Norway. Anyway, and then this is a big European study, the European Alzheimer Disease Biobank. Many people here involved, I guess, and you see it's working there pretty well. And you can see if you have larger, this here we have included more SNPs in the prediction tools. And you have quite a good sort of spread from the highest to the lowest risk. And we do the same with Parkinson, a bit more noisy. We didn't do the same smoothing here, as you see, but the same point with these age-dependent diseases, you can get the hazard scores are working pretty well. So why, Sudha alluded to this earlier, that, you know, why do the prediction? And that is, there's been a lot of trouble, of course, but now there are new drugs available. First of all, they have a lot of side effects, and I'm not sure they're working that well. So, you know, anyway, but there are many more in the pipeline now, and so this is happening as we speak. A lot of these drugs will come the next 5 years. So I think we need to figure out who should have the drugs, and also who should not receive them. So that's why we have another way of predicting, and this is more, I think, relevant for sort of clinical use cases, and that's if you have come to the memory clinic. The other ones I showed was the population prediction. I'm not sure how likely that will be to have that sort of rolled out in real life. But this, I think, is more, again, relevant. Again, you need to start treatment early. And of course, how can you know when you get the disease? Early, before it's the brain damage has happened. If you need to wait for the symptoms to appear to diagnose it. And that is this kind of catch-22 in the field. And here we then added the polygenic hazard score, did this, we worked with Anders Dale, you know, this is, by the way, also some kind of machine learning developed. Do you know FreeSurfer? It's a machine learning tool to estimate the size of the brain structures. And so, because we you know, even though I like genetics, I don't think that's the whole story. No, but I think that's always a problem. Oh, I only work with imaging or only work with genetics. Oh, I'm expert in, you know, cognition. And I think we should combine this, and that's what we've done here. So the question is very relevant clinical question. You come in the door, I have memory complaint. Will I develop Alzheimer's disease? And here is the same modeling approach, but this is when you enter the clinic, and here 5 years later. And if you only do age and genetics, you get a nice stratification on different genetic risk factors. But then the odds ratio here is 3.5 between 20% highest and 80% lowest. You can see the different risk percentiles. And then if you add then imaging, can we then explain more? Can we predict more? If you, and that's, I mean, if you come to a memory clinic, you do imaging. So that's part of the standard of care. And then now we can see there's these groups here with the lowest risk are very unlikely to develop Alzheimer after 5 years, but these are much more likely. And now it's odds ratio of 13. But then the last thing you do in the clinic when you come in and have memory complaint, you do cognitive testing. And if you add that to the prediction, then we get up to odds ratio of 27. And this, you know, these people here, you can say at baseline, probably you don't need Alzheimer's disease treatment, you not have these drugs that are really dangerous. You can instead do, maybe it's depression, maybe other stuff. But here, these are where you should have the action and start interventions. And that's talking about that, even though there are some new drugs available, it's super expensive to run these clinical trials. So what if you do this hazard score? You can also reduce the sample size. So if you go out on the street, then I want to start a clinical trial. And then, oh, let's only do people above 70 or something. Then you reduce the sample size. But then if you add genetics, you reduce it to 50%. And then if you do imaging or cognition, you get down to 1/3 of the total sample needed. And that's really beneficial for drug industry. So much easier to run clinical trials if you can reduce the variation. And then, so this is also something we're working on, because if you want to do all this kind of fancy big data, imaging, genetics, there's not a doctor in the world who will understand anything. You know, it's so complicated. So that's why we're working now with this EU project where we try to develop this kind of risk report integrated with cognition and imaging to have like a portal for physicians to discuss these things, because otherwise I think the Framingham risk factors for cardiovascular disease is a really good example. But then if you want to do, if these kind of polygenic multimodal things are ever going to have impact in the clinic, you need something like this, at least for people like me who are just a psychiatrist who you know, it's really difficult to understand this in clinical practice. But then I have a couple of minutes at the end for some Nordic things we're working on. Because these other things is something we're trying to implement and test. I guess here in the UK, you could also try this in your healthcare system. So the Vikings, you know, I think they even came to London at some point, didn't they? But it's overlapping genetics, you know, similar genetics, and also culture. And I think that is, so we share a lot of the same background and also history. And roughly 27 million total is like London times 2 or something. It's not that big, but anyway, we have these personal identifiers, you know. You're born and then you have this number follow you the rest of your life for everything, healthcare and taxes and everything. We have registries. We can follow everyone in the registries. I can just even now go on my phone and look up when I did my vaccinations, when I was hospitalized and everything. We have the biobanks. We have now, I think, at least 1 million in the biobanks. And we have a public health system it's not Mayo Clinic or, I don't know, Imperial, or what's your clinic here? But it's decent. And we have many of these surveys that have been running in the population, and we have a motivated population. So I think we are working now to see if we can integrate research and healthcare and really make this into like a discovery platform. It's not primary disease kind of biopsy or autopsy verified diagnosis, but we have large numbers and we have national cohorts. And it's a bit cold, so it's not representative for the whole world, but still it can be used as a very nice little kind of discovery platform. And so I mentioned this before, 90,000 here in Hordaland. This on the west, 36,000. This Tromsø, I think there's 35,000 now, all genotyped. And then in addition, oops, can you get this back on here or? Yeah. So in addition, we have the mother, father, and child cohort. And there, I think it's good that we have many young people here because there are 114,000 genotyped children that will develop Alzheimer's, but that will be into the future. But the point is we have prospective data on them and also the parents. So we have now totally like 400,000 all genotyped that are followed in the registries and also with questionnaires. And we're trying to develop this into, we can't share the data. You need to sort of sit in Norway and analyze them because it's really sensitive. But we are combining them and also the healthcare samples into some kind of analytical platform. And it's all federated, so we can also combine it then with, you are using the same ICD code, roughly the same healthcare system in Denmark, Sweden, Norway, and also in Finland. And that's perfect. So now we are running trying to analyze this across all the samples with, I think we have these are genotyped roughly across like neurodegenerative diseases, 25,000 we're analyzing now, and 7,000 Alzheimer's disease diagnosis. And then we can really figure out what are the kind of gene environment interplay that is happening. And then we don't need to do Mendelian randomization or anything. We can have longitudinal data. I think that is what we can sort of try to scale up a bit so we have enough power to find these risk factors. And then what we could do then, and that's the point, if we build a model now, we've got a 4M or something, then every year we'll have new cases coming in. And then it's totally independent. It's not like reanalyzing. And so we can have like a hackathon and sit down and say, okay, did the model work in the how many new dementia cases do we have in 2027 or something? That should be predicted based on all these variants. Anyway, so that's what I wanted to say. So I think making the gene discoveries and Alzheimer's disease have clinical impact is really important. I think the certification clinics is what we can apply now, or just need to validate it and make sure it's useful in real life. I think this multimodal kind of epidemiology approach is really useful. It's not like we have some favorite variables or anything. We just need something that is predictive. And then if we can use the Nordic infrastructure then we can really get up to speed on discoveries and also validation of the findings. And I think this concept of looking for in genetics, then looking for rare variants, that's more into disease mechanisms, but the common variants we should be able to use pretty soon to have impact on this kind of prediction and stratification schemes. So these are where the head of the dementia in Norway is Geir Selbekk, and we work a lot with Anders Dale with this method development. Thank you.
Yeah, it's incredibly impressive. So you had on your in science or something, but it's not open. I mean, UK Biobank is truly open. Sitting at their desk analyzing UK Biobank data.
Yeah.
And I mean, I know you're not going to solve it, but you know, what's your view on open science, making these data available? I mean, this is a fantastic resource. You could, UK Biobank, combine with UK Biobank. At the moment, anyone in the world can use UK Biobank.
Yes. No. So, but I think I totally agree. We have access to UK Biobank, and we paid like $50 or something, or it's incredible. And is it your taxpayer money, or is it? Exactly. So, no, but it's we're trying to convince the government and everything to make it available. But I think there are what we are trying to develop now, and that is really this thing here to make this into a system, because you cannot have the data taken out, and I don't think you can get sequencing data from UK Biobank. That you have to, so that will be no, yeah. So that is the model, and then it is possible. And many people here are also working on like MOBA, but you just need to follow the regulations, then it can be done. So what we are now trying to set up this kind of system here to make it possible for others to get access. But then, as we discussed also, like in Denmark, this country here, they have a lot of data now, and even non-consented. So it's, taking the blood spots, genotype them, and follow them in the registries without asking you. And that is never going to leave Denmark. So that is a different kind of approach. But still, you can team up and analyze, and that is. But what we are trying to is develop this kind of a federated. You can have a container. Anybody working with that, standardized. If you harmonize all the data, and then you can send out scripts and do some IT. You can even do some kind of easy learning, I think, on kind of scripts and get them back and send out again. So it is possible, but it's we at least we had GDPR, that was a nightmare, and all kind of. So we have learned that it's better to try to do it without changing Norwegian law in a way. But
being able to get access, analyze the data. I agree, you can't send the data out. I mean, as you say, with UK Biobank now, with the sequencing data, all sorts of reasons that can't be sent.
Yeah, we have to save the planet. If you have to copy the whole genome sequencing, the temperature.
Yeah, so you DNA nexus, and it costs you money. So yeah, if this could be made available so that people could dive in, get the data out without people, and that would be fantastic.
Yeah, no, but it's fantastic now. But I think, but you talked about earlier about it's so difficult to get funding for just compiling, generating data. And I think this is the same here. Everybody wants to, if you have a new machine or some stupid new method or new, then you get funding. But we want to share data from boring registries, then, no, it's not exciting. So I think that is, but I think it is possible to run some kind of, to set up collaborative grant applications. Okay, you have your specialties here in the UK with all kind of this and that. We have this big data, follow them up, but we just, so I think this I think we just need to make the best out of it, and I've given up trying to convince the government and everything.
It sounds much more exciting to me to develop collaborations than to try to convince the government to. But God, it would be fantastic to do that. Really nice talk. I just wanted to go back to your models, and I was really inspired by your efforts to bring this into the clinic and to make something practical, to make it individual as opposed to population. What I wondered is if you'd considered, or whether it would be important in the next stage to try to think of an age to look at the hazard score and the other factors on an age-related basis, so that you add a third dimension to this. So you look at surfaces, and I'm thinking very much about what one does in pediatrics. You look at head size as a function of age, single metric, and the polygenic risk score should change with age.
It's super important. So I didn't talk about that here, but we are now the big MOPA study where we have children talking about growth curves. Then you can train the model. We have some new, if you do repeated measures, you can build genetically determined trajectories, and you can call it your digital twin, which is a real digital twin. And then you can, of course, like height. Everybody knows genes. You will, for height, you will get there when you are 18, 19 years, but then it's too late. The point is, if you can figure out what genes, what is the age, 5 years of age, your height, because then you can say, oh, you are too small or something, and then you can do intervention. Then you can do something in the clinic. No, but that's no. Sorry, your perfect digital twin say you should be here on your height, but you are here, and it's then you can do something. Then you have to get vitamins or stop drinking that water. So there, but if you end up when you're 19 and you got this height or this, then it's too late, because then why do you need to know how? Well, I will be 1 meter 90 centimeters. And that was correct. You didn't get there. You ate too little, but you need to know when in time you can intervene, and that's why you need these.
Your data set, age of evolution, and it'd be incredibly exciting. It would help people so much. Also just getting away from the confounds of case control at earlier ages.
But it's not perfect, and I just with a chat here. So we spend, so much time and effort building this. So, and it's not like, it all looks so nice, but we're missing data and everything. But there's a lot of opportunities. Like in MOBA, there are 55,000 trios or genotypes. So you can look at, anyway, but they're not yet old enough to develop Alzheimer.
Thank you very much, Ole. Thank you.